we finish with the syllabus, the better. Ne? There's no sort of restriction or any law saying, okay, I should stretch the syllabus up till this many months before the exam. No, the earlier we finish the syllabus, the better. But of course, the way we are covering the syllabus, it's not just we are just reading the resource book. We are making notes. We are doing questions. We are having exams. We are having a paper class that is following up everything. So with our plan, okay, I think we have the proper sort of the perfect balance of theory coverage. So with that, we are anyway going to cover the syllabus on time. Okay, at least my plan is for you guys. Sorry, at least 2 months. Okay, so it could be slightly changed, but definitely, right, at least 2 months I plan to cover before the exam. Okay, because I started your theory in August. So we started a little bit behind, but I think we have caught up sufficiently. Okay, so anyway, so today it's going to be mainly moving on to the physiological functions of the kidneys. Okay, because last week we actually went through all the different excretory systems that different phyla started to develop as they evolved. Okay, as they evolved, we talked about different excretory systems, starting from your flame cells, protonephridia, into metanephridia, into other special things such as Malpighian, and also your salt glands, right? Then we move on to the human urinary system mainly. So even though our main topic is excretory systems, right? So if you think about the excretory systems, our lungs, our skin, they also can be considered as sort of routes of excretion, but our main nitrogenous excretion happens through our pair of kidneys. So that's why we are going to mainly focus on the urinary system in this lesson. So anatomy of the urinary system, I think we completely finished. Okay, starting with the overall, let's say, macroscopic view of a kidney. Before that, how the urinary system anatomy, entire set of organs are arranged in the abdomen as well as the pelvis. Then we moved on to the actual anatomy of a human kidney, right? A slice cross-sectional view. Then we classified nephrons, right? Then we classified nephrons. What are the two main types of nephrons? Okay, I'll switch to the software for a little bit. Okay, everyone can hear me, right? Send me a response. You can hear and see the board, right? Yeah. Any issues? No. Okay. So just to sort of warm up, we'll start with a bit of, I'll show you some slides, okay? So I'll show you some slides so that we can just get into the thing, okay? I have some cross sections. Oh yeah, okay. Right. Okay, so we'll move on to this sort of the 3D anatomy of a sort of this slice of the kidney, right? This slice of the kidney. So what do you call this outer part of the kidney, right? Outer part of the kidney, that means, okay. So actually the kidney has this mainly, it is held in place by a mass of fat, as well as there's a fibrous sort of outer casing, right? There's an outer casing or outer covering around the kidney that is called the renal capsule. So that you can see. So actually the mass of fat can be found outside this as well as inside this. Certain people, due to certain disease conditions, they have a buildup of fat inside the renal capsule as well. So that is possible, right? Anyway, so if you take the kidney, there are two main areas that you can sort of visualize as well. I'll show you a real slice section. There are two main areas you can visualize, right? Outer what? Always outer what and inner what? Outer what, inner what? Whether you come to the kidney, whether you come to the ovary, whether you come to the adrenal gland, usually this nomenclature is the same. The outer area is called what? Outer, yeah, the outer part of the kidney. Outer cortex, still. Outer cortex and inner medulla. Outer cortex and inner medulla, right? So outer cortex has what appearance? I think we drew arrows and said outer cortex has what appearance? Outer cortex is what? Outer cortex is granulated. Outer cortex is granulated due to the presence of what, maybe? Due to the presence of glomeruli. Maybe there's one glomeruli here, one glomeruli here. Outer cortex is granulated due to the presence of glomeruli. Okay. Then when you come to the inner medulla, of course, you can see in this diagram also, but now we have drawn some one or two nephrons. Okay. So the inner medulla has what appearance? Has what appearance? The inner medulla? Sort of a irregular appearance, or irregular appearance. That is what? That is called the striated appearance. So striated appearance is due to the collecting ducts, right? Striated appearance is due to the mainly collecting ducts. You can see one main collecting duct going through this this part of the adrenal medulla, inner medulla. Okay, so this inner medulla is mainly composed of what? Then you can say these individual things that we call as, this single unit is called what? It's a renal pyramid. It's a renal pyramid. So this is the renal pyramid. Okay, so inner medulla is composed of mainly renal pyramids. Several renal pyramids are there. Okay, so now what else? So this is what we call as the renal pelvis. Okay, renal pelvis is where almost all collecting ducts empty into that, right? Like the common drainage point, right? When you have several sort of a kanu coming and emptying into one main pit, right? So that is called the pelvis. So the pelvis goes out of the kidney. So you can see what I wanted to mainly show you in this diagram, the two types of nephrons. There are two main types of nephrons you can find in a kidney. There are short nephrons that are extending a short distance into the renal medulla. Then there are nephrons that are extending deep or long into the renal medulla. Those are called what? Short ones are called what? Yeah. What are the short nephrons known as? Yes, what are the short nephrons that are extending a short distance into the renal medulla? So they are mainly found in the renal cortex. They are mainly found in the renal cortex. Yes, what are they called? If they are mainly found in the cortex, cortical nephrons. Sorry, cortical nephrons. If they are extending into the medulla, juxtamedullary nephrons. Juxtamedullary nephrons. Okay, juxtamedullary nephrons and then cortical nephrons. So, which type of nephrons do you have mostly? Which type of nephrons are usually found in a normal kidney? Which are most common? Yeah, you have small nephrons are the common. Cortical nephrons are common. Do I look like a bit misty looking? I did some adjustments yesterday because the class was dark. Now a bit normal, yeah, okay. Anyway, right. Cortical nephrons and juxtamedullary nephrons. So that is okay. Then we can move on to the structural and functional unit of the kidney. What is the structural and functional unit of the kidney? What I always told you. What do you mean by the structural and functional unit of the kidney? Structural is easy to understand. No? So the kidney is built up of these things. The building unit of the kidney. What do you mean by a functional unit of a certain organ? Any organ, if it's the functional unit. That means if you want to say this entire organ function is done by this smallest unit, that smallest unit that is able to do the function of the big organ is the functional unit. It's independently functional as the functional sort of the organ itself, right? So if you take the kidney's main functions are osmoregulation and excretion. So if there is a certain system that is able to do that function in the kidney, that is the functional unit of the kidney. So if you take one single nephron, it is able to do the kidney's job. Therefore, the nephron is the functional unit of the kidney as well. Right? So we can zoom into a simplified nephron, right? A simplified nephron. So every nephron is supplied by an artery, right? Every nephron is supplied by an artery, and that artery is going to later become a vein, right? You can see that artery is going to later become a vein, and there's a vein going out of every nephron. Correct? There's a vein going out. So the arterial supply first comes into this ball of capillaries. This arterial supply first comes into this ball of capillaries. That ball of capillaries is called what? Glomerulus. The ball of capillaries is the glomerulus, and what is surrounding it? This sort of cup-like structure, that is the Bowman's capsule. Right? So glomerulus is the ball of capillaries. It is surrounded by the Bowman's capsule, right? And then from the Bowman's capsule, you start your other tubular structures of the nephron. Now, so this first part of the nephron that is extending immediately after the Bowman's is called a proximal convoluted tubule. Proximal convoluted tubule is going to continue into what we call as the loop of Henle. So loop of Henle has a descending part. There was a question last week, how do you know which is descending and ascending? The part that is leading, the proximal convoluted tubule is leading into the descending one. Then it takes a 90 degree sort of a rapid sort of a 180 degree turn, and then it comes back up. So that is known as ascending limb. From the ascending limb, you have distal convoluted tubule, which is, you can see, a slightly shorter than the proximal convoluted tubule. That leads into the collecting duct. So you can see the collecting duct has the ability to collect urine from several nephrons. Okay, from several nephrons it is able to collect. Okay, so this is the basic anatomy. What else can I show you? So this is a kidney section. This is mainly something we followed, right? So you can see in this section, you can see the outer cortex, renal cortex, and these are the renal pyramids. Okay, these are the renal pyramids, and what else? So these are the renal pyramids. These are minor calyxes and major calyxes. You don't need to know this. This is the renal pelvis. Okay, so if you hide the renal pelvis, you can see how the inside of the kidney looks like. So from the renal pelvis, you lead into the what? Your urine takeaway tubes. Those are your ureters. You have two ureters. Ureters are a very common place of what? Kidney stones getting stuck. We are going to study about kidney stones as well. Very good place of kidney stones getting stuck. When you have a kidney stone in this one of these ureters, very painful. Huh? Because kidney stones are not like very shiny pebbles you find in the garden that are very smooth. No, kidney stones are very, very rough because they are formed by precipitation. No? They are formed by precipitation and are a growth. Therefore, they have very pointy endings. Very painful when you have a kidney stone passing through this. So the two nephrons, ureters, are going to go into your urine bag, that is your bladder. This is your bladder. It is mainly a smooth muscle walled organ. Right? Then this is a male system of, obviously you can see, right? From the bladder you have a very long urethra in a male. Right? From the bladder you have very long urethra. From this point until it comes out of the penis, you have a very long urethra in a male. I think if you switch to, okay. Kidney cross section we already went about. So just keep in mind, kidneys are not just alone. Hari, I think I mentioned this in my diagram that I drew last week also. Kidneys have this sort of a cap-like structure. Kidneys have this sort of a cap-like structure. That structure we'll very soon talk about when you are doing endocrine system. Right? Those structures, anyone know what are these structures? The cap on top of the kidneys. What are those? Yeah. Adrenal glands. Yeah. Adrenal glands. So they are main part of our sort of endocrine system. They actually have two parts themselves. So adrenal glands are sort of hanging above the kidneys. And so with this I can show you some of the blood supply of the kidneys as well. So I told you the veins, big veins are in front of the big arteries. Okay, so who's the big red artery going down? This is the abdominal aorta that is going down from the aortic arch. Right? So from the aorta you have into the left kidney you have the left renal artery. Okay, into the left kidney you have the left renal artery. You can see, and onto the right side like this, you have the right renal artery. Right? Those are taking blood to the kidneys. Then in front of them you have the renal veins. Okay, so there's a right renal vein and there's a left renal vein. Can anyone tell me what is this big vein? What is this big blue color vein? And the vein, is it, how do we say, is it going upwards or downwards? Yes, it's inferior vena cava. Is it going upwards or downwards? Blood in the inferior vena cava, is it going upwards or downwards? Upwards. Because why? The blood is going to go to the heart, no? From everywhere, from the legs, abdomen, collected, and it's going to go towards the heart. Yes. What else can I show you? I can show you some real kidneys. Yes. This is how a sliced kidney looks like. Okay. So mainly you can identify cortex and medulla, not very clear, right? You can see bit of striations. Can you see? You can see a bit of striations in these areas. Right? Can you see a bit of striations in these areas? Those are your collecting ducts. Okay, those are your collecting ducts. And what else can we see? So you can of course see the renal pelvis sort of here in this area, renal pelvis. What else? So major calyx, minor calyx, you don't need those things. Mainly renal pelvis can be seen. Okay. Yeah. Then there's another kidney here. Yeah, in that you can very clearly see pyramids. You can very clearly see individual pyramids. There's one here, there's one here, there's one here, and there's one here, right? Renal pyramids you can very clearly see. On the other side also you can clearly see the renal pyramids, right? Individual renal pyramids you can see. So the kidneys are very small, okay? Right, of an adult. These are also slightly sort of swollen because they have this whole sort of preserving liquids. Otherwise, the kidneys are very small. Maybe an average size, maybe 5 inches, okay? Not even 5 inches, yes, right? Very small. But the function they perform, they filter out so much blood per day, right? So very physiologically active organ. So this is actually, this is that inferior vena cava, this is the aorta. Okay, then you have the left and right kidneys. Sorry. The entire renal blood vessel system has been separated out. What else? Also in this diagram you can see the two kidneys here and here. Okay, you can see the two kidneys. That is basically an abdominal dissection. Okay. Yeah, and here this is actually a female system. So you can't actually see any parts of the urinary system. This is actually the female uterus. Sorry, this is the female uterus. So you have the fallopian tubes, the two. This is one fallopian tube. This is the other fallopian tube, and the ovary. There's one ovary here, and the other ovary I can't actually see. Okay, right. Anyway, yeah, there's a uterus there, no bladder. Yeah, uterus, a lot of uterus are there. That's it, right? Yeah. This is, so here the kidneys have been sliced from the top. So this is the, hold on, this is the right kidney. This is the left kidney, right? So this is actually the back vertebra, right? What we call as the lumbar vertebra, lower back vertebral column vertebra, right? This. So now can you understand, Harit? Your kidneys are located on either side of the vertebral column. I told you, right? When we are describing the locations of the kidneys, how did you describe? They are located on either side of the vertebral column, right? Either side. Then also behind the peritoneum, you can't actually see the peritoneum. So the peritoneum actually runs like this. So this entire front part are everything located in the peritoneum. So you can see. Kidneys are usually sort of pasted to the posterior abdominal wall behind the peritoneum on either side of the vertebral columns, because this is the vertebral body. This is actually a lumbar vertebra, lumbar vertebral body. Right? So keep that in mind. Very, very commonly asked how to describe the location of the kidney. Yeah. Anyway, so that's enough, no? Any questions? Okay. All right. Any questions? No. Right, come to the resource book. Then we will continue with today's work. Alright, we will continue with today's work. So, okay. Take out the diagram tube as well. Okay, right. So where were we? So did we finish discussing page number 228? Yes, no? So we discussed sort of the microstructure of the glomerulus and the Bowman's capsule, right? So we discussed what is the inner wall of the Bowman's capsule, its composition, and the outer wall, and how the into the capsular space, how the filtrate comes, okay, how into the capsular space, how the filtrate comes, and so we have to continue from there onwards, whatever is going to happen, right? So what is the main thing that is happening in the glomerulus and the Bowman's capsule? What is the main process that is happening? It's a filter, right? It's the filter. It's the sort of a rough filter, right? From what did I tell you? The filtration occurs through two surfaces, right? The filtration occurs through two surfaces. Firstly, right? Imagine this is the capillary wall, right? Imagine this is the capillary wall of what? The capillary wall of the glomerular capillaries. So this is the capillary wall of the glomerular capillaries now, right? You know, capillary walls, they are leaky, because they are composed of simple squamous epithelium. Then of course you have the inner wall of the Bowman's capsule as well. The inner wall of the Bowman's capsule as well. The inner wall of the Bowman's capsule also, so there's a complicated structure there, also have spaces, right? So the urine, sort of not urine, the blood plasma can pass through these two surfaces, through their gaps, into this what we call as the capsular space, into capsular space. So now you have a filtrate, right? So now you have a filtrate in the capsular space, okay? So loop of Henle, PCT, DCT, everything we have done. So today we move on to page number 229, process of urine formation, alright? Process of urine formation. Let's read. Main steps in urine formation. There are 3 steps involved in urine formation. Let's read. They are ultrafiltration, selective reabsorption, and secretion. Okay, alright, let's read. Ultrafiltration is the filtration of blood under high pressure into the cavity of the Bowman's capsule. Alright. Filtration of blood under high pressure into the cavity of the Bowman's capsule is called ultrafiltration. Can you highlight? I think we finished ultrafiltration also last week, neither? Anyone? Did we finish ultrafiltration also last week? We did, no? Didn't we make a note also in under the glomerular? Yeah, we did, no? Yeah. Yeah, can you send to the main chat? I'm missing your messages. Okay, hurry. Right, so ultrafiltration done. So quickly tell me then, if I told you this filtrate, hurry, if I told you this filtrate, now that you have in this capsular space, if I tell you, is it equal to blood plasma? Am I right or wrong? If I tell you this filtrate that is present in this capsular space, if it's equal to blood plasma, am I correct or wrong? Somewhere correct, ne? But what's missing, right? So not exactly the same, right? If I remove some certain things, so I can say blood plasma minus few things, right? Blood plasma minus blood cells, plasma proteins, and platelets, okay? Then the filtrate is equal to blood plasma, okay? Blood cells, blood platelets, so platelets are not considered as cells, no? Platelets, cells, and large proteins such as plasma proteins, if I remove from blood plasma, then it is equal to this filtrate, okay? Then it is equal to this filtrate that you have in the capsular space of the Bowman's capsule. So now exactly tell me what are the things that are being filtered, right? What are the things that are found inside this filtrate? What are the things that are found inside this filtrate? Quickly revise with me. Yes. What are things? Water mainly. Hari, water. Certain ions, usually a lot of ions, calcium, sodium, potassium, right? So many ions are there. Amino acids are filtered. That is very bad, okay, because they have to be recovered. You have to recover your amino acids, amino carbo protein stick, okay. Then glucose, almost all glucose is going to be filtered, okay. Then vitamins are going to be filtered, usually water soluble vitamins, sorry. Then nitrogenous phase, those are the things you have to filter, no? Nitrogenous phase. What else? And other small molecules, certain drugs that have been broken down, drugs that have been metabolized in the liver that are now in the bloodstream will be filtered. Okay, right. So those are the things in the glomerular filtrate. Right now, where are we right now? Here, at this point in this nephron, we are at this point. So now you have a glomerular filtrate at the capsular space or at the end of the Bowman's capsule. So now what we are going to do is we are going to recover certain things as we go along this other parts of the the nephron, as well as, so one thing you are going to happen is you are going to recover certain things, right? Things you need. For example, amino acids, your glucose, your vitamins, those things you need, no? You can't let them go out in the urine. So you are going to recover certain things, that is known as reabsorption. You're going to recover certain things. And at the same time, certain things that didn't get filtered at the small amount of time that they were remaining in the glomerulus, you have to forcibly put them in the urine, right? You have to take them and put them in the urine. So that process is called as what? You are excreting certain things, right? Excretion. From the blood capillaries surrounding the nephron into the nephron, there will be certain things that are sort of excreted or forcibly put in, right? So those are the other two processes that's going to happen sort of in the consequent parts of urine formation. Hari? Okay. Okay. So let's keep reading the resource. Moving on to selective reabsorption. Okay. The process through which useful molecules, ions and water from the glomerular filtrate are recovered and returned to the interstitial fluid and then to the capillary network of the tubules is called selective reabsorption. Can you highlight? They can ask this as a definition. What is selective reabsorption? Okay. They can ask as a definition. Useful things, useful molecules, ions and water from the glomerular filtrate. Because glomerular filtrate, keep in mind, it's a bit of a rough filtration. There's nothing sort of actively deciding what should be filtered. Based on the size, they are going to get filtered. Based on the size, right? Based on the size. Big molecules are not going to be, big molecules and cells are not going to get filtered. Small molecules, almost all are going to get filtered. Small molecules, ions, right? Those are going to be filtered. Nothing very selective about glomerular filtration. Okay. So what are they saying here? Return into the interstitial fluid and then into the capillary network. So can you understand? Now imagine, if we are talking about the proximal convoluted tubule, around this proximal convoluted tubule or even the distal convoluted tubule, you have a capillary network, a supply of blood. What is that capillary network called that is surrounding this DCT and PCT? Yes, I think we label, maybe this box we wrote down a name. What is it? So if this green box is proximal convoluted tubule, what is the capillary network that is surrounding both the PCT and DCT? Yeah, capillary network. Okay, what is the capillary network surrounding this? Peritubular capillaries. Very good, very good. Okay, peritubular capillaries. If you didn't label this last week, can you label, Hari, in your tubes? Peritubular capillaries, right? Then so the other part, so it's the same network, but we divide it into two parts, right? So surrounding this distal and loop of Henle's, right? Sorry, descending and ascending limbs of loop of Henle's, you have another capillary network. They are all part of the same network, but two main areas, okay? These are known as vasa recta, right? Vasa recta. Two capillary networks are there. So now imagine when you zoom into this, okay? Whenever you want to reabsorb something, imagine you have some glucose molecule here. Alright, when you want to reabsorb that glucose molecule, it has to come out into the sort of the tissue fluid. Tissue fluid surrounding this nephron, and then from that tissue fluid into the blood capillary they have to go. So that's what they are saying there. Okay, so into the interstitial fluid, when you are doing selective reabsorption, it's going to go from the tubule into the interstitial fluid into the capillary. Okay, keep reading. Most of the reabsorption from glomerular filtrate back into blood takes place in the convoluted tubules. Sorry, most of the reabsorption takes place in your DCT, sorry, PCT mainly, then in DCT as well. What are the things? Ions such as sodium, water, and valuable nutrients such as glucose, amino acids are absorbed either actively or passively from the initial filtrate. Yeah, there are so many different types of transporters. There are active transporters, passive transporters to absorb certain things. These mainly your nutrients, okay, things you need, right? Oh, Kilivella, you are going to absorb them. You should not allow them to go into the urine. Otherwise, if you have glucose in your urine becomes very sweet. Okay, that's how early doctors used to diagnose diabetes. Okay, tasting the urine. Okay, right. Okay, secretion. Secretion is the other part of urine formation. Right? Process by which foreign material and substances not required by the body, including waste, are cleared from the peritubular capillaries and interstitial fluid into the filtrate is called secretion. So secretion is the opposite of this. Right? Things that didn't get filtered, but you have to get rid of, that are found within these capillaries, they will be first sent to this interstitial fluid, and then from the interstitial fluid they will be put into the nephron tubules, okay, with the tubule of the nephron. Got it? So you are clearing things, okay. So keep reading. Secretion is required. Such substances may not be entirely filtered due to the short time they remain in the glomerulus, right? So glomerular filtrate, I think I told you what is the average rate of filtration in the glomerulus? About 120 milliliters per minute. 120 milliliters per minute, so that comes to about 180 liters per day. Right, getting filtered in the glomerulus. So it's getting filtered fast in the glomerulus. So in the short amount of time, there's no time for certain things to get removed. Okay, therefore that's why you need secretion. They can ask you, okay, can you highlight, not a definition but an important point. Why do you need secretion? Page number 200 and where are we now? 230. Can you see? Secretion is required. Certain substances may not be entirely filtered. Why? Because of the short time. Because of the short time, they go through the glomerulus very fast. They remain in the glomerulus. Okay. Substances that are secreted into the filtrate include H+, ammonia, creatinine. Creatinine. Highlight that list. Okay. Drugs such as penicillin and aspirin. Okay. So many other drugs are there. But these are called renal excretion. Certain drugs, I mean, almost all drugs are renally excreted, but some drugs are, let's say, not broken down directly in the kidney. Some drugs are directly broken down in the kidneys and excreted. Some drugs are broken down in the liver and their metabolites are excreted in the liver. Sorry, kidney. So it doesn't matter. Kidney is the main place where you have the excretion happening. Right? And excess potassium. Right? The tubular secretion of H+ and ammonia are important to maintain the normal pH in the blood by the formation of ammonium in the urine. NH3 can combine with H+ to form ammonium. That is actually what we call as a buffering action. This H+ and ammonia combination, right? Have you do buffers in equilibrium, ne? Chemistry, the biggest lesson, final one. Equilibrium, you do something called buffers. Buffers are usually weak acids or bases. Okay, so ammonia is a weak base neither. So it's going to combine with H plus and to become buffer, right? So NH4 is going to be less acidic, right? So H plus is directly acidic nature. If you have H plus, that is directly acidic. So ammonia is going to sort of catch that H plus and is going to make the ammonium ion. So with that, acidity will be regulated. So it's a buffering action, right? There's a buffering action by phosphate. So many other things are bicarbonate buffering action also there. But anyway, this is the one that is mentioned here in your research. Okay. All right, we'll summarize all those things later at the end as a one full note in this model nephron that I have given you. All right, so let's keep reading. Okay, so now we are moving on to, in your resource, one of the most classically a lot of information packed into just one page. That sort of situation you can remember, right? Him pages in your resource where there's a lot of information just packed into one page, very high yield pages in the resource. So we are going to encounter such a situation. Okay. Secretion occurs in the proximal and distal convoluted tubules. Secretion may be either active or passive depending on the location and the substance. Yes. Sometimes you need some energy in order to excrete certain things, secrete certain things. Okay. So for that, sometimes you might need some energy. Okay. Right. Let's go. So we are going to mainly talk about the PCT now. What's going to happen in the proximal convoluted tubule. Okay. So we can put an arrow and write here. Right now, what do you have here at this point here? You can say here earlier if you, I think earlier we wrote that what are the composition of glomerular filtrate we wrote last week. Right? So you can put an arrow. This is glomerular filtrate at the end of the Bowman's capsule here. So now we are going to focus on up to here. Usually up to here, this is the PCT, proximal convoluted tubule. So we are going to focus what's going to happen in PCT. Okay, so let's read. Glomerular filtrate in the Bowman's capsule, which contains all the substances in the blood except blood cells, platelets, and large molecules, passed into the PCT. At this region, selective reabsorption of ions, such as sodium, water, and valuable nutrients from the initial filtrate occurs. Okay, so one of the most important things that is happening in PCT, there's a sodium chloride co-transporter that is actually an active transporter. Okay, that is going to be working in the proximal convoluted tubule. Okay, yeah. So one of the most important things that is going to be reabsorbed here is your sodium. Alright. So sodium of course sodium he has this clingy friend that is chlorine. Okay, chloride ions are going to come as well. So when sodium and chloride are coming, that means it's a sort of ions are moving out of the tubule. Ions are moving out of the tubule. As ions are moving out, what's going to come? Together with the ions moving out, because of the shift in the water potential, H2O is going to come out as well, right? A lot of water reabsorption is going to occur. All right, let's read. Then what are the valuable nutrients such as glucose and amino acids are from the initial filtrate occurs? Nutrients, especially glucose and amino acids, are actively transported into the interstitial fluid. Yes, for glucose reabsorption as well as amino acid reabsorption, there are specific transporters. So I'll use some red color. Glucose is reabsorbed. Then amino acids. Amino acids are also reabsorbed. Okay, so amino acids are also reabsorbed. Right. Then cells lining the tubule actively transport sodium into the interstitial fluid, and this transfer of a positive charge of the tubule drives the passive transport of Cl-. Proximal tubule also reabsorb K plus and most of the bicarbonate by passive transport. Alright, then there's another two set of molecules that are going to be reabsorbed here. I'm going to mention with this color, right? There's going to be K plus and bicarbonate HCO3 minus. So whenever you are making this note, use some colors. Okay, use some colors. Otherwise, both of us will not be able to understand what we have done. Okay. So K plus and bicarbonate is going to be also reabsorbed in the proximal convoluted tubule. So what we have been talking about is this is all reabsorption, right? Selective reabsorption. Okay. So keep reading. The reabsorption of bicarbonate. Where are we? Okay. K plus and more to the bicarbonate by passive transport. Reabsorption of bicarbonate into the filtrate contributes to the pH balance of body fluids. Yes. Bicarbonate helps to maintain, usually it sort of swings the pH towards the alkaline direction. Bicarbonate usually swings it towards the alkaline direction. Okay. So helps to maintain pH balance. So depending if your body is acidic, if your body fluids are alkaline or acidic, the bicarbonate reabsorption can be sort of regulated as the body requires at this point. Okay, so it's not a fixed amount, you can control it. Okay, as solids move from the filtrate into interstitial fluid, water is reabsorbed by osmosis. Very important. Water by osmosis. Because you know water travels, water is a solute, no? Sorry, solvent. Water is a solvent. So that when the water moves by the concentration gradient, we give it a special name. So it's not diffusion at that point. When the water moves by sort of a concentration gradient, you have a special name for that. No? So in plant form, we learned that is called osmosis. So water gets reabsorbed by osmosis. Okay. A major portion of water reabsorption occurs. Yes, actually most of the water reabsorption occurs here in the PCT. Okay. As filtrates pass through the proximal convoluted tubule, secretion of specific substances into the filtrate takes place. Right. So now reabsorption is done. Right. So we have taken out the things we want from the tubule. Now it's time to put things in the tubule that we don't want anymore. Put things in the tubule that we don't want. What are the things we are going to put? Firstly, we are going to put excessive protons. Excess protons we are going to put in the tubule. Okay. Where are we? Okay. As the filtrate passes through the PCT, secretion of specific substances into the filtrate takes place. The cells lining the tubule secrete H plus by active transport. So this is an active transport into the H plus by active transport and ammonia by passive transport. So H plus and who's his friend? Ammonia. So ammonia will use some sort of this cathode green, right? This green. This is NH3. Something important is going to happen within the tubule. Okay, so these two are going to combine here. The ammonia and the H plus is going to combine here. So I'll use this sort of gray color. And what are you going to get? You are going to get NH4 plus. NH4 plus is going to be produced within the tubule. Alright, NH4 plus is going to be produced within the tubule because of the combination. Secreted ammonia act as a buffer to trap NH plus, forming ammonium, NH4 plus ions. In addition, some materials such as drugs, toxins that have been metabolized in the liver are actively secreted into the lumen of the PCT. Right. So that's the end of PCT story. So there's going to be some secretion of certain drugs as well. So I'll use this pink color, right? Certain drugs are also secreted into the proximal convoluted tubule. So PCT it's a place of many, a lot of things are happening in the PCT. A lot of things are happening in the PCT. I think it's better if you take this down after I have drawn everything, no? Otherwise, it's going to be a mess. Okay, please can you highlight the things? I couldn't tell you to highlight as we go along. Can you highlight? Glucose and amino acids are actively transported. Can you highlight? Glucose and amino acids are actively transported. So mena me denna actively reabsorbed, okay, actively reabsorbed, okay, glucose and amino acids, and sodium is active, okay, sodium occurs by active transport, okay, sodium occurs by active transport, sodium active reabsorption, okay, sodium active reabsorption. So keep in mind when sodium is reabsorbed, the chloride comes passively. When the sodium is reabsorbed, the chloride comes passively because of the individual, because it's going to follow that positive charge. No? So chlorine, I'll remove this. So chlorine is going to follow passively. Okay, chlorine is going to follow passively. What was this here? Yeah, you have K plus. Right? Passive water and also, sorry, passive chloride and water by osmosis. Right? Highlight that and reabsorb most of the bicarbonate and most of the K plus by passive transport. In the resource, can you highlight this? So these two guys, these are passive. K plus and bicarbonate, they are passive. Okay. Water reabsorption we talked about. Okay, then H plus. So we move on to secretion. Secretion of H plus occurs by active secretion of H plus. Okay. Then ammonia is passive. Ammonia secretion is a passive process. Okay. Ammonia secretion is a passive process. Then what else? Drugs are also actively secreted. Drugs are also active. You need energy, like you have to sort of forcibly remove them, right? Somehow get out, Kela, you have to remove the drugs forcibly. Okay. So I think after we have drawn this entire diagram, we'll write down these things in your diagram. Otherwise, it's going to be a bit of a mess. Okay. Right. Any questions? Now we have completed PCT. Right. Actually, big portion of this discussion is over. Where is the filtrate now? Where is the filtrate now? At this point. The filtrate is now at this point here. Proximal convoluted tubule is over. Now we are moving on to the loop of Henle. Particularly in the loop of Henle, the descending limb of loop of Henle. Okay, let's read. As a result of water reabsorption and secretion of different substances, the filtrate becomes concentrated as it passes through the proximal convoluted tubule. Yes, right? So what happened? So at the end of each of the parts of the nephron, you should be able to mention whether if the filtrate, so at this point we are mentioning the fluid that is traveling through this nephron as the filtrate, right? So you have to be able to say, has the filtrate become more concentrated or has it become more diluted? So they say it has become what? What? It has become more concentrated loop. Why do we say? So compared to the filtrate here at the end of the glomerulus or Bowman's, the filtrate at the end of PCT, they say it's more concentrated. Why? Two things. One thing, you reabsorbed or you took out the water, or you remove the water, that is going to concentrate it. And the other hand, you put more solutes into it. No? You put ammonia, or you put H plus, you put certain drugs, right? Because of those things, the filtrate is now a bit more concentrated than it was at the end of Bowman's. Okay, clear? All right. Now we move on to the collecting ducts. Collecting ducts. Let's read. Sorry, I'm sorry. Moves into loop of Henle. As the filtrate moves into the descending limb of loop of Henle, passive reabsorption of water through osmosis continues. So for this, I want you to just let's read this. There's a nice principle, okay, happening around this loop of Henle. It is something nephrologists are very obsessed with. Nephrologists are people who have this diagram in their bedroom wall. They like the nephron very much. Nephrologist. Anyone? Okay. Anyone whose parents are nephrologists? No. Okay. Right. So there's something very interesting happens. Your loop of Henle. Now listen carefully. Your loop of Henle is the place where you produce your concentrated urine. Most of the actual water reabsorption happens, and sort of the water recovery is sort of mainly focused here. So there's a principle that is going to do that water recovery, that is by working. The descending limb as well as the ascending limb of loop of Henle are going to work together to do this. Okay. So for that we need to have the entire story with, we have to read the entire story. Okay, let's see. As the filtrate moves on to the descending limb, passive reabsorption of water through osmosis continues and the filtrate becomes more concentrated. Filtrate reaches the ascending limb of loop of Henle via the tip of the loop. So this is the tip of the loop, right? So water is there. Sorry, the filtrate is there now. Ascending limb is impermeable to water. Can you highlight that? We already know that. Ascending limb is impermeable to water. We already know that, okay? Ascending limb is impermeable to water. So that no water reabsorption takes place, but a considerable amount of sodium chloride reabsorption occurs. Highlight no water reabsorption where at the ascending limb, ascending, going up. No water reabsorption takes place, but a considerable amount of sodium chloride reabsorption occurs. Most of the sodium is reabsorbed into the interstitial fluid by active transport. Okay, yeah, most of it is active transport because there's a certain active transporter. If I can remember, sodium, some sort of a transporter is there, right? So now listen. Okay, may first focus on this ascending limb, the limb going upwards. Okay, let's focus on this ascending limb or the limb going upwards. The main thing that is going to happen in this ascending limb, because of a specific transporter, sodium, and of course together with sodium, who's the friend of sodium, and as well as chloride are going to be recovered or removed or reabsorbed from this ascending limb. Right? Because why? The ascending limb is impermeable to water. So water can't go out. So what's going to happen? Now listen. This, so usually below this point, may point again below this point usually is found within the medulla of the kidney, inner medulla. Usually your loop of Henle is sort of found within the medulla, right? Loop of Henle is usually found within the medulla. So because of this sodium and chloride reabsorption, this medulla is going to stay what we call as hyperosmolar. Hyperosmolar means what? Again, this I'm talking about this, not the kidney nephron, but the surrounding of this nephron. The surrounding of this nephron, it's very salty. The surrounding of this nephron is very salty because of this sodium and chloride reabsorption. The surrounding is very salty. So because of that salty nature, so with using your knowledge, you can say this outside has a very low water potential. Because why? Outside solute concentration is high, no? Solute concentration is high because of this sodium and chloride reabsorption happening, particularly in the ascending limb. Surrounding this loop of Henle, the solute concentration is high. So when the solute concentration is high, water potential is less, no? So when the water potential is less, because the descending limb is the only one who's permeable to water transport, water is going to come out of the descending limb. Correct? Water is going to come out of the descending limb. This is a continuous process. When filtrate is coming down, water is going to go out because of this high solute concentrations in the medulla. So I'll mention this. This is the medulla. This is the renal medulla. So medulla is very salty, we say. Kidney kalabalan depa. You can't taste the kidney, but we say the renal medulla is very salty because of this mainly sodium chloride. So as the ascending limb or the limb going upwards is impermeable to water, there's no water reabsorption happening. So the only place water reabsorption can happen is the descending limb. Okay. So that is the main story of the loop of Henle. Okay. So we can say this is the descending limb, this is the ascending limb, right? So you can mention here impermeable to water, or you can say water impermeable better, no? Water impermeable ascending limb. Okay. Ascending limb is water impermeable. This is the descending limb, this is the ascending limb. Okay. Right. Where are we? So, okay. So sodium chloride absorption happens in the ascending limb. Okay. Where are we? Yes. At the end of using NaCl but not water, the filtrate becomes more diluted as it moves towards the distal convoluted tubule. Correct? So now listen. Water or the filtrate at this point, right? The filtrate at this point, what can you say? You lost water, no? Filtrate at this point, you lose water, therefore it is more concentrated. At the tip of the loop of Henle, it is more concentrated. But now, when it is about to enter into the distal convoluted tubule, what can you say? Because of losing the solutes. So, at the tip of the loop of Henle, it became more concentrated because you lost water in the descending limb. Now, in the ascending limb, you are going to lose solutes. So, at this point, now again the the filtrate is more dilute. Right? As it goes into the distal convoluted tubule, it has now become again dilute. Okay. So at this point also we can mention at the end of the proximal convoluted tubule, right? And the end of proximal convoluted tubule you can mention more concentrated, no? Because why? You lost water as well as you secreted certain things. No? So this is the story of the loop of Henle. This is the story of loop of Henle. Okay. Clear? Okay, very annoying. Okay, hold on. I want to remove these lines and add a different color to show you that its high solute concentration is there. Otherwise, it's a bit weird. Okay. Okay, it's impossible. Hurry anyway. Right. So now clear what is going to happen in the loop of Henle? What's going to happen in the loop of Henle? Clear, right? So water reabsorption happens in the descending limb and solute reabsorption is going to happen in the ascending limb. Therefore, urine at the end, it's going to become more dilute at the end. Okay, so we can say this is the medulla. Okay, this is the medulla with the high solute concentration. Okay, so this is the medulla. Anyway, so we can move on to DCT, distal convoluted tubule. Alright, let's read. The distal convoluted tubule plays an important role in regulating K plus and sodium concentration of body fluids. The amount of K plus secreted, okay, the amount of K plus secreted by active transport and the amount of sodium reabsorbed from the filtrate can be varied at the site according to the needs of the body. Right? Can you highlight? Plays an important role is regulating K plus and the sodium concentration of body fluids. The amount of K plus secreted. So always you are going to get rid of K plus, right? So always you are going to put K plus into this, okay? K plus secreted, okay, by active transport. K plus secreted by active transport as well as sodium reabsorbed from the filtrate. Okay, so sodium is going to be reabsorbed. So sodium is going to come out. Sodium reabsorption, it is also active. Hari, it is also active. Can be regulated at this site. Hari. Distal cannula tubule also contributes to pH regulation by a controlled secretion of H plus. So you can secrete H plus here as well. Okay, so you can secrete H plus into here as well. H plus active. Then also you can secrete bicarbonate. What color did I use for bicarbonate? Also purple. No? You can reabsorb bicarbonate at the distal convoluted tubule according to the needs of the body. Okay. At the distal convoluted tubule, passive water reabsorption can be increased under the influence of antidiuretic hormone. So we'll talk about antidiuretic hormone with a lot of detail. Alright, so H2O can be controlled, it can be increased or decreased, we'll say under ADH, antidiuretic hormone, that can happen in the distal convoluted tubule. Then there's another hormone we need to know, aldosterone. I'm going to explain all these because if you go to the next page of the tute, you can see we have a, this is mainly your antidiuretic hormone. No wait, this is the antidiuretic hormone related mechanism. This is the aldosterone mechanism. Right? So both hormones we are going to make a note separately. Okay, so don't worry about that. This is the antidiuretic hormone mechanism. This is the aldosterone mechanism. But just for now, remember there are two main hormones that are going to be acting here in the distal convoluted tubule. Okay, one is antidiuretic hormone ADH. Okay, aldosterone secreted by the adrenal glands stimulates to increase reabsorption of sodium and water and the excretion of K+. Okay, so aldosterone has an impact on sodium and potassium. The filtrate of the distal convoluted tubule finally leads to the collecting duct. Okay, so we can add here. Aldosterone is going to somehow influence this. Okay, so we'll add here. Aldosterone can have a impact on here. Okay, aldosterone can have an impact on this active secretion of potassium as well as active reabsorption of sodium. Aldosterone. Okay, all right. Then we move on to collecting duct. Final part. Okay, urine formation almost done. We are going to do some questions and I'm going to somehow slightly summarize it very soon as well. Okay, very quickly as well. As the filtrate passes along the collecting duct, filtrate becomes more concentrated and urine is formed. Now at this point, we stop calling this as filtrate. We stop calling this as filtrate. We can now call this properly as urine because at this point you can understand, no? Right? Water has been reabsorbed, right? Drugs are being introduced, right? Then glucose and amino acids have been recovered. You have put more ammonia into it, right? So at this point, there is something very similar to urine that you put in the washroom. Okay, right? So this is very similar to urine at this point. Few more changes to be done, and you have proper full urine. Okay, collecting duct. Okay, so let's read. As the filtrate passes along the collecting duct, the filtrate becomes more concentrated and urine is formed. At this site, water reabsorption can be increased under the influence of ADH. So, in the collecting duct also, our antidiuretic hormone has a big impact. Here, ADH, H2O, absorption can be increased or decreased under the ADH here in the collecting duct as well. At the collecting duct as well. Aldosterone hormone stimulates the active reabsorption of sodium and passive reabsorption of water in the collecting duct. Okay, so aldosterone has an effect on here as well. Aldosterone can have an effect on the collecting duct as well. Aldosterone. There it is going to regulate. Okay, we'll better mention it like this. So in the collecting duct also sodium reabsorption can happen. Right? Sodium reabsorption can, Sodium reabsorption can happen. We are going to take down small notes for each of these, okay? So don't worry about it, okay? Sodium reabsorption can happen in the collecting duct as well. That is under the influence of aldosterone, okay? That is also can be controlled by aldosterone hormone, okay? That also can be controlled by aldosterone hormone. Sodium reabsorption happening at the collecting duct. So because of course with the so active reabsorption of sodium, what's going to happen? Water is going to come passively. Okay, because osmotically active molecule is going out, no, therefore water is going to follow. Right, water is going to follow. Because of the high urea concentration in the filtrate at this region, some urea diffuses into the interstitial fluid. Yeah. So at the collecting duct area, there could be some passive. It's a bit uncontrollable that some of the urea that you find within this collecting duct can come back into the interstitial fluid without our control. Without our control. You can't see this, no? This color you can't see, no? Right? What the hell? Okay, right. It's very annoying. Okay, right. So sodium reabsorption is under aldosterone influence, and some amount of ammonia, as I was saying, some amount of ammonia can be passively reabsorbed. Again, it's not something we do voluntarily, but because of the very high concentration of ammonia at this point in the collecting duct, some will sort of escape. Some will sort of escape into the interstitial fluid. Okay. Final processing of the filtrate at the collecting duct forms urine. Okay. So, what is the output product you are getting? Urine. Urine. Final output of this factory is urine. Okay. So, you can understand, you can have a nice summary of the entire nephron here at this point in this page. Okay. We are going to make some more notes at the bottom edges of the page as well. Okay, so let's see. Can you start taking this down? Okay, can you start taking this down? I'll send a photo or screenshot to the groups as well. Okay, leave some space here at this side and at this side to take down some notes. Okay, and but more than that, you don't need to worry. So go with the order. Go with the order. Start with the proximal convoluted tubule, then move on to the collecting, loop of Henle, then into distal, and then collecting duct. Okay, I'll send a screenshot of this as well to the groups. Okay, but I think it's better if you can go with location by location. Okay, so start with the PCT and let me know when you have completed the PCT. Okay, anyone who's not able to see clearly, you can see, no? Unable to see? Let me know. Can see. Okay. That's a way I can take a screenshot. I don't know how have I. Okay. Okay. You can see a bit more right now, eh? Okay. Right. If I zoom out, you can't see entire thing clearly, no? If I zoom out, this is a bit unclear, eh? Yes. Bit unclear, eh? Okay, so let me know when you are done with proximal convoluted. Let me know when you are done with proximal convoluted. Then I'll sort of go down into the collecting duct. Sorry, loop of Henle. Okay, let me know. Okay, wait until I take a full photo so that if you miss anything, so then you can complete at your free time also. Wait, until I take a photo. Okay, urine becomes more concentrated at the bottom. That part also needed. No? Okay. Okay, I'll zoom in now. Let me know when you want to see the loop of Henle. Okay, let me know when you want to see the loop of Henle. Okay. Let me know when you want me to scroll down. Okay. So while you are going through this, sort of pay attention to active or passive. So I didn't go with the color coding or something. I didn't go with any color coding to show you active or passive. I just went with the different colors for different substances. Okay, so I thought it's better to write active or passive right next to it, because I mean when you have this sort of a active and or passive නිකන් color code එකක් වගේ එකක් තිබ්බම you have to use your brain a bit more. Okay, this is active color, passive color. Better to just write there whether it's active or passive, no? Okay, that's what I thought. Okay, it depends on your preference. Okay, you can use a color code for all the actively absorbed things and also the passive absorbed things, but that's going to make it a bit more complicated, I think. Okay, so make your notes as simple as possible. Okay, right. Okay, so keep in mind also the direction of the arrows. Okay, direction of arrows, if they are going towards, if they are going into the tubule, that means it's an excretion or a, sorry, secretion. Okay, and if they are coming out, that means they are a selective reabsorption. Okay, keep that, pay attention to that also. Then don't forget to sort of mention the influence of ADH here, right? ADH in the distal convoluted tubule as well as the collecting duct. ADH has an effect, okay? Antidiuretic hormone, okay? So keep that in mind as well. And the effect of aldosterone in the active absorption of sodium as well as the active secretion of potassium of that both those things aldosterone has an effect. So keep that in mind when you are writing this. Aldosterone, two effects, okay? Okay. Can I scroll it down so that you can write what is there in the vasa recta? Sorry, the loop of Henle? Yes, what do you think? Okay, shall I just zoom it out like this? Okay, right. Then I'll make small, just a small addition here. We'll put a small star mark and write here, right? You guys, nothing much, okay? So we'll put a small star mark and write here. Sodium reabsorption can only be regulated naturally in the collecting duct via aldosterone hormone. Just one point there. Sodium reabsorption can only be regulated naturally in the collecting duct via the hormone aldosterone. Okay, that is an important point that you better remember. Okay. Method I forgot to add. This urea is passive, right? This urea reabsorption that is happening in the collecting duct is passive. We don't want that urea. We don't want that urea, but it's happening because it's very concentrated at that point, right? Because it's very, very concentrated. We can't control. It's going to get reabsorbed. Okay. Also, very carefully at the end of this, at the tip of the loop of Henle, the urine or the filtrate is more concentrated. You have to add that. Okay, all right, sorry. That's it. No need to write anything here. All right, no need to write anything in this edge. Okay, you can use that space if you want. Okay, got it. That's it. So after you have drawn this, we'll take a small break, five to at least 10 minutes. Okay, can we take? Okay, we'll take around 10 minutes. And what does A and D denote in the medulla? What do you mean A and D? This is descending limb. This is the ascending limb. Are the descending limb? Okay, so the arrows are wrong. Okay, sorry, the arrows are wrong way. This is the descending limb. Okay, just to mention that. Okay, this is the descending limb, and the other one is the ascending limb. Okay, take a small break, and after the break we'll discuss about aldosterone and antidiuretic hormone effects. So I think I told you to have the grade 13 resource book with you as well, right? Because we are going to jump to a bit of grade 13 work there. So it's important that you have the grade 13 resource. If you don't have it, at least get the PDF right now. And so we'll take a small break. Okay, small break. 10:45 with the everyone join. Okay. Okay. Quickly come back. Take a small break and come back. Okay. Okay, all right. Shall we continue? Yes. Anyone who needs this to be written down? Okay. So yeah, if you try, you can make this a very neat note. I who's actually sent one of the notes that some of you might have made actually. It's really interesting that you can use a lot of colors. And that's why I kind of wanted all of you to be at home so you can find some color pens and do this nicely. Because this is the first step to sort of memorizing this whole process. It starts with proper organization of the information. Harish? It has to start with proper organization of information. Otherwise, this is such a it's like such a long paragraph containing so much information. Okay, so the first thing we have to do is to make our lives a bit more interesting, makes bit of some sort of an attractive note. Note means a bit of a let's say this sort of a image that's going to show you the full summary of what is going to happen, the big picture, what's going to happen, and at the end of this phase, this phase, this phase. Okay, right. Okay, we'll continue then. Okay. Okay, right. Let's continue. So we have to next move on to a discussion of a different regulation. Several, actually, two regulatory mechanisms you need to follow related to the renal system, kidney regulation, but they are a bit complicated, and with that, I prefer we take a full big picture understanding together with the endocrine part associated with that. Okay, together with the endocrine part associated with that. Otherwise, it's a halfway job, I feel like. So what I want you to do is, so this is the simpler one, but we'll first start with the aldosterone related mechanism. Okay, aldosterone related mechanism. I'll screen share for you, and so anyone who doesn't have the grade 13 resource book, anyone who doesn't have the grade 13 resource book, yes, anyone? Okay, what I want you to do is, can you go to page number 42? I'll screen share and then we'll read it together. Okay, so in the grade 13 resource book, page number 42, you can hear me, right? Okay, all good? Okay. I'll screen share for anyone who's not able to, was not able to find it. Okay, so we'll quickly go through it. There's a part of osmoregulation in grade 13 resource under our homeostasis. Okay, so yeah, let's just go through the resource with me. I'm screen sharing for you now. Okay. When there is osmotic balance, the amount of water and concentration of solutes is the same inside and outside the cells. Osmoregulation is important for organisms to keep a constant optimal osmotic pressure within the body. In the humans, osmoregulation ensures that total blood volume and the concentration of dissolved substances in the plasma and tissue fluids remain constant within favorable range. One phrase that I want you to pay attention here, of course, you know what is osmoregulation, that is maintaining water and solute balance. But the other word I want you to use is menomega, blood volume. Blood volume is such an important phrase, right? The amount of blood that is flowing through your blood circulatory system at a given time. So you know, basic hydrodynamics. Yes, basic hydrodynamics, you know. blood circulatory system is a closed system. No? Within the blood vessels, it's a closed system. So inside that closed system, you have a circulatory fluid, right? So because it's a closed system, it is under pressure. Right? Imagine because of some this volume of circulatory fluid inside the blood circulatory system increases, right? So there's a pump that is pumping this circulatory fluid around, but because of this volume increase, what's going to happen? The volume of the blood has increased, but the volume of the circulatory system is the same. So that when the heart is exerting the same amount of force because of the increased volume, there's going to be increased sort of force of blood being ejected out of the heart, as well as because of that, the pressure that blood exerts as it travels is going to increase. Right? So whenever blood volume increases, blood pressure is going to increase. So keep that in mind. Whenever blood volume increases, blood pressure is going to increase. Okay. Right. Let's move to next page. In the human body, osmotic valence is achieved in two ways. Right? Read. Controlling the amount of water and controlling the amount of solutes gained and lost by the body. Right? So either one way is, so if you take blood as a solution, you can either control the solvent of that solution. No? What is the solvent of that solution? That is water. Then what is the so what are the solutes? Mainly what you can control by osmoregulation is your ions, no? Your ions. So either those, in any one of those two ways, you can control osmotic balance. Blood water homeostasis is controlled by the hypothalamus. Okay, so hypothalamus, I will show you the location in the brain. That's very soon we are going to actually discuss. Okay, if you really want to see what is hypothalamus looks like, Hari, you can come to our diagram tute, and so you can come to, this is hypothalamus, Smithy. Hari, this is hypothalamus. So in the main, so the brain sort of, there's a hanging part, so there's a thalamus, and so that's why we call it hypothalamus. So it's located below and in front of the thalamus. We call it hypothalamus. Hypothalamus is this. So hypothalamus is actually it's an organ composed of several nuclei, right? It's like several small coordination centers all collected into one organ. Okay, so then it has a sort of a very closely associated organ that is called the pituitary. Pituitary has two parts, as the anterior and posterior. Okay, so anterior and posterior pituitary is there. So hypothalamus, so the pituitary is like a servant of hypothalamus. Pituitaries are like a servant of hypothalamus. According to the orders given by the hypothalamus, the pituitary is going to do some certain things. Okay, come back to the resource. Right. Hypothalamus contains osmoreceptors. Osmoreceptors are receptors that can detect osmolarity. That means increase or decrease in ion concentration, or increase or decrease in water balance. Okay, which can detect osmolarity of blood passing through the brain. In response to the osmolarity or osmotic pressure, so osmotic pressure is the sort of the physical measurement of osmolarity. Okay, so we say when osmotic pressure increases means, that means osmolarity has also increased. Again, you have a lot of solutes. You have a lot of solutes and water concentration is reduced. We'll take a note. Okay. Osmoreceptors. So can you highlight, if you have the grade 13 resource, we'll highlight it together. Hypothalamus contains osmoreceptors which can detect osmolarity of blood passing through the brain. Okay, so not exactly. So that osmoreceptors, they act as the main sort of the input for the hypothalamus to control our osmotic pressure. Okay, in response to the osmolarity or osmotic pressure of the blood, the hypothalamus controls the sensation of thirst. So now we are tackling this first part. Okay, hypothalamus controls the sensation of thirst. Thirst means what? Tiba, the feeling that you get that when you need a drink. Drink means usually water. Right? So thirst and the generation of secretion of the hormone ADH. ADH anti-diuretic hormone. Diuresis means, when you think of the word diuresis, it refers to sort of losing water, dehydrating wage idea එකක් තමයි, that is there in the diuresis. So antidiuresis means you are opposing losing water. So you are not losing water, that means antidiuretic effect is done by the antidiuretic hormone. Okay, right, let's read. So this is this paragraph is from this paragraph is from about antidiuretic hormone. Let's read. Okay, when blood osmolarity is increased beyond physiological limits, it is sensed by osmoreceptors in the hypothalamus, which stimulates the posterior pituitary to release ADH into the blood circulation. Can you highlight? Osmoreceptors stimulate posterior pituitary, very, very important. The part of the pituitary that we say sort of is found behind, stimulates posterior pituitary to release ADH into blood circulation. Okay, right. ADH acts on the kidney tubules and stimulates the reabsorption of water from the distal convoluted tubule of nephrons and collecting ducts. Very important statement. Hari. So this is actually the disorganization of your resource book. Hari. That feels like several people who have no communication with each other wrote the resource book. Hari. Because in the excretory systems, there is no mention of any this is not properly explained under excretory systems. But the proper word by word explanation is given in the endocrine system. So how are you supposed to learn this when you are doing excretory system? So that's the issue. So can you highlight? ADH acts on the kidney tubules and simulates reabsorption of water where DCT and collecting ducts. Water reabsorption can be controlled under the influence of ADH at distal convoluted tubule and collecting duct, producing concentrated urine. Now, concentrated urine means what? How do you see if your urine is concentrated? You make more yellow urine. More yellow urine. The yellow color to the urine comes with a bit of ammonia in the it can come, and mainly because of sort of a red blood breakdown product, red blood cell breakdown product that is called urobilinogen. Urobilinogen, that is yellow color. So when you have a little amount of water in urine, that yellow color is more pronounced. Can you understand? When the urine is more concentrated, there is not enough water to dilute that yellow color. So your urine becomes very yellow colored when you don't have, when you make what we call as concentrated urine. So when you have ADH being secreted by the posterior pituitary, you make more concentrated urine. Okay. When the blood osmolarity is decreased, ADH is not secreted. So water reabsorption through the distal convoluted tubules of the nephrons and collecting ducts stop, thereby producing diluted urine. So when the blood osmolarity comes to normal level, ADH secretion is going to stop and blood osmolarity, blood osmolarity is now normal. Okay, so you are going to start making as usual sort of normal urine. That means you can call it as dilute urine as well. So that is the story of ADH. Now let's come to what is given in your resource book under excretion. So this additional section, I think it was added after this revision of the resource book. Refer osmoregulatory section of grade 13 for further details. So that's what I did for you. So this flow chart is there. Osmotic pressure increases. Okay, blood osmotic pressure increases, maybe because of so many things. Because you had a very salty meal. You had some dry fish or bacon or whatever, something very salty you had. Okay. And or you didn't drink water for a long time, or you sweated, you produced a lot of sweat. Okay, you did a sport, or you were in a very hot environment or whatever, you lost water. So now your blood osmolarity has increased. So now that will be detected by the osmoreceptors in the hypothalamus. Can you highlight? It will be detected by osmoreceptors in the hypothalamus. Hypothalamus is going to respond in two ways. You can see it is going to respond in two ways. One, The simplest mechanism is it's going to generate thirst. It's going to generate the sensation of thirst. Therefore, you are going to drink more water so that drinking water directly can reduce the blood osmolarity. So, by increasing the intake, you increase your water intake. I told you blood osmolarity can be controlled in two ways. One way is by controlling the water intake. Second way is by controlling the solute intake and excretion. No? So, two ways you can control, because osmolarity, blood is a solution. No? So, either you can control the solute or the solvent. So, here we are controlling what? The solvent. You are adding more solvent, that is water, so that you can dilute it. Right? Second way, the complicated way bit, okay. Hypothalamus is going to send a signal to his servant, who is the posterior pituitary. You are going to secrete the hormone ADH. Can you highlight? Increased secretion of ADH. Okay. You are going to increase the secretion of ADH. Kidneys. ADH is going to go and act on the kidneys. Increase reabsorption of water by the distal convoluted tubule and collecting duct. Okay. So, very important to remember, where is the water reabsorption happening? Distal convoluted tubule and collecting duct. Okay. So, have an idea about the normal osmotic pressure as well, about 300 milliosmoles per liter. Milliosmoles per liter, that is the unit of measurement of blood osmolarity, milliosmoles per liter. So, nephrologists are very concerned about this blood osmolarity value. Whenever to the ward, the nephrology team comes to refer a patient, that's the first thing they ask. What is the osmolarity? Okay. Anyway, okay, clear. So, now jump to the question tube. Diagram tube. Okay. Then we are going to make our own flow chart with memory. Hari. Did you see there's a kidney stone? Yeah, randomly I have put a kidney stone. You can see how sharp and pointy it is. Ne? Okay. So, right. So, this page is dedicated to our osmoregulation. Okay. So, I want you to divide it into exactly, not exactly, sort of somewhere here. You can divide it into two. Okay. So, we'll write down the topic. Okay. So, try to draw a much straighter line than me. Okay. Then you can write here the topic. Regulation of blood osmotic pressure and fluid retention by antidiuretic hormone. Better if you can write the full name, antidiuretic hormone. Okay. So, any sort of regulatory mechanism, now listen, any sort of regulatory mechanism in your body starts because a certain parameter means what? A certain condition. So, in here it is the osmotic pressure or osmolarity. It could be your blood glucose level, it could be your blood pressure, it could be your body temperature. Some sort of a condition or a parameter in your body changes. That's going to act as what? A signal. So, that's going to act as a signal. That signal is going to be what that's going to start this process. Okay. So, what is going to be the signal that is kick starting this whole process? What is going to be the signal that is actually starting this entire process? That should come into this gray box. No, what is the gray box? Anyone? What should we write in this gray box? Hold on, there's a power cut. Hold on 1 minute. Yeah, what should come in this gray box? Yes? Anyone?
something that is kick-starting the entire process, right? Something that is kick-starting the entire process. So what is that? Yes, blood osmolarity increases, no? Right? So here we can write blood osmolarity. We'll say, okay, we'll say increase in blood osmolarity. Increase in blood osmolarity is going to act as the signal. No? Increase in blood osmolarity, we can say, no need to know the normal value here. Okay, so we can write here increase in blood osmolarity. Okay. So that is going to be the signal, no? Alright, so now you have a problem. Something has changed in your body. Something that should be maintained within a normal sort of parameter has changed. Okay, so now you have a problem. You need to regulate it. Sorry. So that is going to act as a signal. Okay, so that is going to act as a signal. So we can indicate that it's acting as a signal to the what? Who is here? Yes. This is the hypothalamus. This is the hypothalamus and it is going to be detected. So, we will add here, actually it is better if we can add osmoreceptors in hypothalamus detect the increase. Then what is going to happen? Yes, it does two things, no? So, it does two things. One thing here, the simple mechanism generates thirst. Alright, so in hypothalamus there's a special, so I told you hypothalamus is like a bowl of regulating centers. It's like a bowl of regulatory centers. Actually, your sexual behavior, the time you wake up, and your autonomic nervous system, how are you going to react to certain stressful scenarios, your hunger and your thirst are all regulated by the hypothalamus. So, hypothalamus is like your involuntary mechanism controlling main center for controlling involuntary mechanisms. Alright. So, there's a center called actually for generating thirst. Any ideas? It is called thirst center. Alright. Thirst center in the hypothalamus is going to generate thirst. Alright. Because of the thirst, okay, the person is going to drink water. So, we can say drinking water reduces osmolarity. Drinking water reduces osmolarity. Right? Drinking water reduces osmolarity. So that is the simple mechanism. Right? So that is a simple mechanism. The other mechanism is going to involve our ADH. No? Here, hypothalamus is going to send a signal to where? Who is this? Where we can call him as anterior pituitary. This is the anterior pituitary. Anterior pituitary is going to release our main hormone involved in this process, antidiuretic hormone. Right? Antidiuretic hormone is going to go and act where? It's going to go and act on two parts. Right? Very, very important. Please remember the two parts. It's going to go and act on the collecting duct as well as make out the distal convoluted tubule. Alright, distal convoluted tubule. What's going to happen in the distal convoluted tubule? Reabsorption of water. You are going to reabsorb water. Water reabsorption increases. So, both these things, you can either reduce the amount of water you are losing, or either you can increase your water intake. Two things you can do. You can either reduce your water loss, that's what you are doing by increasing water reabsorption, or you can increase your water intake, that's what you are doing by drinking more water. Correct? So at the end, we are working towards what? To reach normal blood osmolarity. Correct? You are going to reach normal blood osmolarity. What is it? 300 milli OSM per liter, milliosmoles per liter. Okay, right? So that is basically it. Okay. Right. So can you come to page number 5 of the diagram tute and divide the page into this half section, and can you start writing this? Okay. So I'll just write down a small note that you can write here. Okay. So once you have done with it, take it down. Okay. I'm just going to write it here. Okay. Hypothalamus regulates blood. Okay, you write this. Okay, write this, and later you can write this down. Regulate blood osmolarity in two mechanisms. Sorry. Okay, regulates in two mechanisms. Production and release of antidiuretic hormone, and second one, generation of thirst. So hypothalamus has two mechanisms that are involved in regulating osmolarity or blood osmotic pressure. Method, I would like you to write something like, shall we say, produce dilute urine. Can you write down? Produce dilute urine. Not because of drinking water, also you can get a bit of dilute urine, but you get dilute urine because of the action of antidiuretic hormone. Okay, produce dilute urine. Produce dilute urine. Okay, so think in your mind, so think or sort of focus on this as you are while you are doing this. Okay, focus on the mechanism itself, otherwise it's useless. That's why I'm trying to do this as a worksheet based learning. Okay, worksheet based. Any questions you can ask? Okay. Okay, so today's there's only one more response to be done. Okay, done. Okay, right. One more mechanism is left, right? Anyone who's still writing? Providing the recording. Yes, I'll be providing the recording. Yes. Doesn't get urine more concentrated. Shit. Right. I'm sorry. Yes. You produce concentrated urine. Okay. I'm sorry. I'm sorry. Yes. Can you please correct it? Yeah. Small mistake there. Yes. Can you please correct it? Produce. Here, when water reabsorption increases, the urine becomes concentrated. I'm very sorry. Okay. Produce. Methana, can you correct it? Produce concentrated urine. Okay. Yeah, logically you can understand, no? When you reabsorb the water in the urine into the interstitium, right? The urine water available in the urine into the interstitial and then into blood, the urine becomes more concentrated, no? That's why I actually chose the yellow color to show you that the urine becomes more yellow. Okay, so yes, it produces concentrated urine. Okay, you get concentrated urine. Okay, sorry. Her. Okay, so the next mechanism that we have to focus is our aldosterone mediate. Okay, so for today we'll talk about aldosterone and we'll be done for today. Okay, so aldosterone, I'll again screen share for you guys what is there in the grade 13 resource. Okay, under endocrine, sorry, osmoregulation. Okay, all right. So next, let's see. So we talked up to here, right? How ADH, so when blood osmolarity comes to normal level, ADH secretion is going to reduce, so that you are going to produce dilute urine. Now keep reading. In addition, low blood volume and low blood sodium ions stimulate the kidneys to produce angiotensin 2. Very important. Okay, so now the signal for the start of this regulatory mechanism is going to be reduced blood volume. Reduced blood volume is another way of saying the blood pressure has reduced as well. Blood volume has reduced means the blood pressure has reduced. So, and the other thing is low blood sodium. So, I told you water and sodium are in a very sort of a close relationship. Low sodium is an indicator that the body is dehydrated. Okay, low blood sodium is an indicator that the body is dehydrated. So specifically in your kidneys, we have an apparatus called juxtamedullary apparatus. Okay, sorry, yes, juxtamedullary apparatus. There, it's a sensor. It's a sensor that is designed to detect those two parameters. That is, low blood volume, low blood sodium will be detected by a sensor in the kidneys. Keep in mind, the sensor is now in the kidney. You produce something called as angiotensin 2. Angiotensin 2 actually comes from a series of chemicals, starting from angiotensinogen. Okay, so we'll come there again. Okay, angiotensin 2 is the sort of the working molecule or the effector final molecule. Angiotensin 2 is going to be actually produced after a series of reactions. So let's see. Which stimulates the adrenal cortex to secrete the hormone aldosterone. So adrenal cortex. We'll talk about what are the parts of the adrenal gland. Just keep in mind for now. the adrenal gland has a cortex as well. The adrenal gland has a cortex as well. That adrenal cortex is going to secrete the hormone aldosterone. Easy to remember, no? Adrenal gland, aldosterone. Aldosterone has these effects. It stimulates the reabsorption of sodium ions by the kidney tubules, accompanied by water retention. So I told you, whenever you have a sodium being reabsorbed or sodium moving in one direction, water is going to follow as well. Whenever you have some sodium ions moving, water is going to follow. Okay. Which is accompanied by, okay, thereby increasing the blood volume and blood pressure, hence kidneys play a role. Okay, right. So that's what we know already. Then we will jump to what is there in the excretion resource book, this diagram. Okay, don't mind it, it's a bit of a mess that I have made with a note. Okay, right. So where is it? Here. Blood pressure or blood volume drops due to dehydration or blood loss. Right? Sensors in the kidney. You don't need to know what is exactly the sensor found in the kidney. Just remember the sensor is found in the kidney and it's going to detect the drop in blood pressure. Blood pressure means it's a decrease in blood volume actually, because you have lost water, you are dehydrated. Okay. So you are going to secrete an enzyme from the kidney that is called renin enzyme. It is having an enzymatic action. Renin is going to kickstart the process. So now you can remember something. This is something similar to blood clotting. So one is going to act as an enzyme to another reaction. Okay. So liver, so you can see how many organs are involved, no? Right? Liver is going to produce a chemical or a molecular substance that is called angiotensinogen. So you know always nogen is referring to inactive form. Nogen refers to inactive form. Crypsinogen, chymotrypsinogen, right? Always inactive version, pepsinogen is called as ogen, right? So with the sort of the suffix this ogen, okay? So that inactive form is going to be converted enzymatically by renin into angiotensin 1. Correct? Angiotensin 1. Then you need another enzyme that is called as angiotensin converting enzyme. Okay, another yet organ is going to be involved. You don't need to know this. It's actually secreted by this angiotensin converting enzyme is actually secreted by the lungs. Okay, someone completely unrelated. It's going to be secreted by the lungs. Correct? Lungs are going to, so this enzyme from the lungs, angiotensin converting enzyme, is going to convert angiotensin 1 into its final form, which is angiotensin 2. Okay. Angiotensin 2, as I told you, is the active molecule, active substance, which is going to have so many effects. One thing, okay, we'll follow the short path. Short path is, it is going to have a direct effect on arterioles. It is going to have a direct effect on arterioles. So you can, basic hydrodynamics, no? When you reduce the diameter of an arteriole or a blood vessel, it's the speed of the travel of the fluid through that is going to increase. So arterioles are going to constrict, blood pressure is going to increase. That is the direct immediate effect of angiotensin 2. The other effect of angiotensin 2, it's that it's going to go and act on the adrenal gland. From the adrenal gland, you are going to release aldosterone. Aldosterone is something we call as a mineralocorticoid hormone. Don't think about it that much. When we are doing endocrine, you will understand. It's a mineralocorticoid. Then the action of aldosterone is what? Sodium and water are going to be reabsorbed in the distal convoluted tubule. Therefore, blood volume, blood pressure is going to get increased and finally it's going to return to the normal level. So this entire process is going to stop. Okay, clear? Okay, so we'll come to the diagram tute again and we have another flow chart to complete. Okay, this, right? So this system is actually called as renin angiotensin aldosterone system. RAS scale, RAS, sorry, renin angiotensin aldosterone system we usually call it, right? So RAS, let's write. Mainly we are mainly focusing here on we can say blood pressure, but because I mean, so we are mentioning here we are regulating blood volume and pressure because that is what's detected by the kidney. The kidney mainly detects the blood volume and the pressure. So if you come to the previous mechanism of ADH that hypothalamus related, the hypothalamus detects osmolarity because hypothalamus has osmoreceptors. Correct? Hypothalamus detects osmolarity. So that's why we say it is a regulation of osmolarity. So whatever you are detecting, you are regulating. No? Correct? So here also, even though we say it's we are regulating blood volume and pressure, it is again something very closely associated with osmolarity. No? Because why are you having low blood volume? Because you are dehydrated. Definitely your osmolarity has also increased. Right? But you are mentioning you are regulating blood volume and pressure because that's what you are detecting in the kidneys. Right? By renin, angiotensin, sorry, by renin, angiotensin, aldosterone system. That is called RAS, RAS system. Right. So let's complete this. So what is going to be the problem? A change above a fixed level, no? A fixed point, something is going to change, right? So we can say blood pressure or blood volume drops. Blood pressure, blood volume drops could be due to, you can write the examples, easy to remember, no? Dehydration in the වෙන්න පුළුවන් or due to excessive hemorrhage, right? Hemorrhage could be due to traumatic hemorrhage, again because of an injury you can have blood loss, or in certain conditions such as what, you can have fluid leaking. Fluid leaking can happen in what sort of dengue hemorrhagic fever. Dengue hemorrhagic fever, you can have a drop in blood volume. Alright, now we move on to the sensation part. Okay, sensation part. So this is, you can just for fun, you don't need to know this. මෙන්න මේකට කියනවා juxtaglomerular apparatus. No need to remember. Okay, ජොලියට දෙන්නේ. Okay, juxtaglomerular apparatus in kidneys detect drop in blood pressure or volume. Right? That is the sensor, right? Then juxtaglomerular apparatus is going to release the enzyme that is the kick starting point of this entire process, renin. Okay, JGA is going to release renin. Renin is going to do what? There's a liver involvement. Now liver is going to produce something, the inactive form, angiotensin. Study with me. Hari මාත් එක්ක දැම්ම පාඩම් කරන්න. No need other time. Okay, angiotensinogen. Renin is going to convert angiotensinogen into angiotensin 1. Angiotensin 1 will be converted into angiotensin 2 by another enzyme. You can write here angiotensin converting enzyme. Angiotensin converting enzyme is going to convert angiotensin 2 into the active form, which is a very important. The active form is called as angiotensin 2. Angiotensin 2 is going to have a main direct effect. So we can write the direct effect here. What is the direct effect of angiotensin 2? You can say angiotensin 2 arterioles constrict. Arterioles constrict. arterioles constrict. That is the one effect. Then who is this? Who is this yellow man? When me lump එකක් වගේ තියෙන, who is this yellow lump? Adrenal gland. Okay, so label him as adrenal gland. Adrenal gland. Then angiotensin 2 acting on adrenal gland is going to release what? Aldosterone. Aldosterone is going to bring everything back into normal level. What is that? Normal blood pressure and volume. Okay, this is the story. The story of aldosterone mediated regulation. Okay, right? Can you take this down? Blood pressure or blood volume drops due to dehydration or blood loss. JGA in kidneys detect the drop in blood pressure or volume, release renin from the liver. Liver, label this as liver. Hari, mea liver ega. Okay, liver produces angiotensinogen, will be converted to angiotensin 1. Angiotensin 1 convert to angiotensin 2 by angiotensin converting enzyme. Angiotensin 2 is going to go and act on the adrenal glands or has a direct effect on the arterioles. Aldosterone from the adrenal gland is going to wait, there's one important thing. What is the action of aldosterone? I couldn't write. Sorry, methana aldosterone. The action of aldosterone we can write. What is it going to do? Reabsorb more sodium and water in where? Distal convoluted tubule, DCT. Correct? Okay, right. Okay, quickly take this down. Okay, done. Anyone who's still writing? Okay, done. Right. So I think we can do a few questions. Yeah, from the question tute. Do we have any excretion related? Okay, right. We can do question number 1. Taggala Kanban. Question number 1. Can you do quickly? Okay, send me the answer. Question number 1. Quickly do this and send me the answer. Structural parts of the human nephron and some processes that occur during urine formation. So it's actually question number 2 neve metana. So there's a small. So you have to match A, B, C, D with P, Q, R. Okay. Okay. Right. Yeah, what's the answer? What do you think? Yes, what's the answer? Okay. Okay, so let's see. So it's you require a bit of let's say thorough knowledge for this. Okay, so secretion of K plus. That means you secrete, or you sort of put K plus into the filtrate that is traveling, no? So even though K plus reabsorption can happen, K plus reabsorption can happen in the PCT or PCT, but secretion of K plus only happens in one place, ne? Secretion of K plus only happens in one place, that is distal convoluted tubule, no? So that is something very unique. Then reabsorption of bicarbonate. For reabsorption of bicarbonate, you can actually take PCT or DCT both, ne? You reabsorb bicarb in both, right? PCT and DCT both, reabsorption of bicarbonate can happen. Okay, so let's see. Then what else? Reabsorption of water. For that, definitely what you can't take is ascending limb. Ne? Ascending limb definitely you can't take for water reabsorption because it's impermeable. So for this, you can say either descending or PCT, no? Either descending or PCT you can say. So if you take those kind of answers, so let's see, R with, so R you can always match with, R you can match with, descending means B, A or B, right? Then you can match Q with either A or D, okay? Then you can match P with only D, right? P and D only matching, there's only two answers. So in there, A and R is false, so that the best answer is answer number 3. Ne? Best answer is answer number 3. Okay. So can you write this as a small review? Secretion of secretion of K plus is DCT only. Reabsorption of bicarbonate, PCT and DCT both. Reabsorption of water, descending limb ke ladda, descending limb of loop of Henle. Okay. Both places you have water reabsorption. Okay. Okay. Right. Can we do 11 and 12 and we can finish for the day? Okay, 11 and 12, quickly do this. 11 and 12. Okay, 11 and 12. Yes, what's the answer for 11? Yeah, what's the answer for 11? Which of the following is are found in the urine of a healthy person, right? Of a normal healthy adult person. Yeah, you can excrete your H plus. No, protons are excreted. pH balance you excrete. Amino acids. That means you are losing protein. No, proteinuria. Actually, proteinuria is found in people with advanced kidney disease or diabetes. Okay, diabetes related kidney disease or whatever. Creatinine, yes. Creatinine is a waste product that is produced due to breakdown of creatine in your muscle, which is a type of energy releasing molecule. So, creatinine can be found. So, creatine is the molecule, energy releasing molecule. It will be broken down into creatinine, which is a metabolic waste. So, when you are breaking this down, you release energy. You can write this down there if you like. Okay. Potassium, of course, excreted. White blood cells, no. White blood cells are not excreted because they don't get filtered at the GFR. Sorry, glomerulus. You can write here, get filtered at glomerulus. Do not get filtered at glomerulus. Okay, right? Creatine, okay? Creatine is broken down into creatinine. When that happens, you release energy. Question number 12. In the proximal convoluted tubule of a human nephron, which of the following are actively reabsorbed? Sodium. Yes, sodium together with sodium and chloride. Sodium is actively absorbed, then chloride follows. Potassium actively reabsorbed. What do you think? Potassium, is it actively reabsorbed? Yes. What do you think? Potassium, is it actively reabsorbed? No, ne? Potassium is passively reabsorbed. Ne? Potassium is passive. Okay. Potassium is passively reabsorbed because actually this sodium and potassium is driven by the same co-transporter. Okay. So, when sodium is absorbed, potassium also comes. Amino acids are reabsorbed actively, yes. Glucose actively. Urea you are not going to reabsorb. So, it's ACD type answer, neither. Right? Of course, you don't need the urea. Right? You are trying to excrete the urea. So, you don't need to reabsorb it. Right? Okay, I think so. We'll continue in the next class. Okay, so we have certain pathologies related to the kidney health to be studied. Certain pathologies, including kidney stones, chronic kidney disease, kidney failure, and those kind of things to be done. Then the next class we'll be starting nervous systems. Okay, we'll be starting nervous system, which is a big part. Nervous system, nervous regulation, and generation of nerve impulses, synapses, and then we'll move on to sensory structures. Very, very important part. Okay, so we are going ahead with the theory with a sort of the usual pace. Okay, so it's up to you to be in touch every day with whatever that is happening in the theory class. Okay, do not get accumulated. That's the main thing you have to avoid. At least be familiar what you have done. Highlight the resource. If you have any doubts in the notes, fill in the blanks in the notes. Okay, do some questions, do some past papers, as well as do your own self studying, either writing the resource or either highlighting or just reading it and memorizing it, right? do your self studying, do not get accumulated. At least do one time of self studying before, together with the theory class. Go with one round of self studying together with the theory class. That is the best way, otherwise it is going to get accumulated. So don't let that happen. Okay, so we'll see. Okay, then any questions do you have? Anything to clarify? Any questions? Okay, so paper class will be, there will be a holiday for the paper class this Friday because ISM is closed. Hold on, no. This Friday, the paper class is completely online. Okay, this Friday paper class is completely online. We are discussing the August paper. We are discussing the August full paper this Friday. Okay, so join with that this Friday at, we'll start at 4:30 p.m. 4:30 p.m. onwards, we'll be discussing the August full paper. Okay. So all physical people who are joining, don't come to ISM anyway because ISM is closed, and the 4th week of any month is dedicated to discussing the full paper that we gave out at the first week of the month, and so be ready for that discussion. Okay, so September month full paper will be released in the first week of September, and I think September month paper class schedule I sent to the group. Okay, so be thorough with that and also prepare for upcoming topics in the paper class. Be completely thorough. Okay, right? So today we'll finish it to here, right? Because I took this online session as a sort of, even though the institutes are closed, I just wanted to complete this section because it's much easier with the smart board as well for you to quickly complete the notes. Okay, right? So we'll stop for the day. Then any questions you have, you can ask right now.